Hello all. As mentioned in class, the part about when we do human genome program, I'll be referring to some of the documents that were initially presented. We discussed this in class, and I mentioned that okay, for the first one, 9195, it starts off with an executive summary, tells you a little bit of a history, because all this will be repeated later. And then, but off note, a little bit of a summary on the scientific goals. 5-year goals have been identified for the following areas. 1 is mapping on sequencing of the human genome. 2, mapping and sequencing of model organisms. Data collection and distribution, the ethics and legal issues, and also to increase the number of personnel, research training, and then also to be able to transfer the technology developed to other areas of research. So in the I've also mentioned that if you have downloaded an earlier copy, please download again, because this one have highlights. Okay, in the introduction, again, it talks a little bit about the history and how the plan for the Human Genome Initiative came about, and in which one of the earlier program goals include you need to make a genetic map of the human genome, high resolution genetic map, and then this is followed also together with a physical map. I hope you're clear between the differences between a genetic map and a physical map. For the physical map, it's not just for the human chromosomes, but also for the model organisms. Then of course you need to sequence it, both human as well as model organisms. And then also what's also important is to be able to develop capabilities for collecting, storing, distributing, and analyzing the data produced. And I mentioned also that initially when they started, and right after they started, they realized that the technologies that were available at the time was not sufficient, and whatever information at that time was not sufficient. So one of the initial phase, they decided that their work has to be done in phases. So they started off with expanding the human genetic map. From what they already know, they need to increase the resolution. That means they need more markers spaced more evenly across the genome. And then, of course, as mentioned, physical maps. That means the 2 physical maps that we talk about is one, having the overlapping sets of clones to represent regions of the human chromosome, and also to place or order the markers so that it can be useful as a reference. And then part of it also is to relook at the sequencing strategy, because using the automated cycle sequencing, you can only read about 2,000 bases per day from one machine, and that cost about $2 to $5 at that time. So they need to increase the speed as well as to lower the cost. Yeah, reading on, these are the 5-year goals, and then we look at the scientific goals. It's divided for you can read through. Here again, it talks about the genetic map and then the need for markers. This explains what is a centimorgan, and then the goal is to complete a fully connected human genetic map with markers spaced at an average to 2 to 5. It says 2 to 5 centimorgans. Later they'll say, you know, they can actually do a lot more. So I think in the subsequent years they decided that they needed 1 every centimorgan. In fact, they achieved much more than that. This is the part where they where they said there are 2 types of physical map. One is to have an overlapping set of cloned DNA segments, and then the other one is also, of course, to obtain the sequences. This was a time when PCR was initially invented and used. So these are some of the techniques I've mentioned in class. So here, a challenging but reasonable goal for physical mapping research projects is to extend the length of DNA segment that can be covered by a single contig. I think at that time when they started off, they can have a contig of about 40 to 50,000. This is mainly because of the vectors. The cloning vector initially was just the plasmid, or even perhaps the cosmid, so that makes the cosmid takes about 40 to 50 kilobases, so that means the length of coverage is pretty limited. Later you'll see that they have developed the BACs, the bacterial artificial chromosomes, as well as the YACs, the yeast artificial chromosomes. This part talks about the STS system. STS are sequence tag sites. Again, a good site to see this again is at the NHGRI. I mentioned that if you go into the educational resources and you look at some videos on mapping, then it will tell you about how the STS are being used. STS basically is a group of landmarks on the chromosome that can be used for identification purposes. So the 5-year goal: assemble STS map for all human chromosomes. Marker space at approximately 100,000 base pairs. I think they've achieved much more than that. They have one marker every 20,000 after about 5 years. And then generating overlapping sets of cloned DNA. So this is what we talked about, the libraries. Sometimes you call it the chromosome specific libraries. Sequencing, this is the part of the document that talks about the sequencing capabilities back then and how they need to improve, reducing the cost as well as coverage. And then okay, here and then there's also this bits about model organism. They will sequence the E. coli, the yeast, the Drosophila melanogaster, the C. elegans, and the mouse, the lab mouse. 5-year goal: preferred genetic map of mouse genome based on DNA markers. These are different organisms that it will be useful for comparison in terms of structure as well as function. And in this part, data collection analysis, again, they have to develop a proper and advanced tool for collection, storage, and distribution. Genome databases will need to be comprehensive and always updated. And if there are several databases, for example, at that time you have the DBSTS, DBEST, even the Genome Data Bank, there's DDBJ, DNA database of Japan. You've got the Gene Bank. You know, all this will have to be integrated and it have to be managed and curated. Develop an effective software and database design to support large-scale mapping and sequencing project. Create database tools to provide easy access, up-to-date physical mapping, genetic mapping, chromosome mapping, and so on. Develop algorithms and analytical tools can be used for interpretation of genomic information. And of course, this talks about the ethical and legal considerations. So they'll have to organize discussions and working groups to address and anticipate implications of individuals and society of mapping. We always talk about the right not to know and the right to know and all this and related issues. Develop policy options that would ensure that information is used for the benefit of individuals and society. So 5-year goal: develop programs addressed at understanding the ethical, legal, and social implications, the LC, of the Human Genome Project. Identify and define the major issues and develop initial policy options to address them. E, F, and G is about to train more people because they need more scientists, and also to ensure that all this work within the scope and within the correct using the correct approaches, that is usable, that the technology is transferable. That means, you know, the same different labs use these different technologies and will get the same results that can be shared. Technology development and also technology transfer. Technology transfer also means that whatever you learn technology wise for the human genome should be transferable to other crops and so other organisms. Okay, all right, so that will be the quick view of the 91 to 95 Human Genome Initiative document. See you in the next one. Bye.